So hi, everybody. I think we can get started with the introductions. We have a few. So it is my absolute pleasure to welcome everybody to the Frisbee Foundation visiting lectureship. Before I introduce our fantastic speaker, Dr. Vijay Sankaran, I would like to introduce Dr. Ed Snyder, who will introduce the Frisbee family who is sponsoring these amazing events. So Ed, Dr. Snyder is professor of laboratory medicine and pathology, and is now the blood bank director at Bridgeport Hospital. We had the pleasure of working with him for many, many years here at this campus. He is still attending in the blood bank and apheresis section at Yale, and he's also doing many things that are keeping us running and honest. So he is a chair of the Conflict of Interest Committee, chair of the Yale Cancer Center Membership Committee, and also of the Data Safety Monitoring Committee. So Dr. Snyder has worked with the Frisbee family for a long time, since his lectureship started in 1998. So welcome, Dr. Snyder, and over to you.
Thank you. Most of you probably don't know much about the Frisbee, so I'll give you just a brief background. In 1988, Rick and Christine Frisbee's son, Richard, developed acute leukemia, excuse me, and he was the first bone marrow transplant in Connecticut, transplanted by Joel Rappaport, who started the program, came down from Boston, as many of our faculty have. And the transplant was not successful. They were going to do a second transplant from his sister, but he passed away before that could be done. So Richard asked his parents to please do what they could to prevent this from happening to other people. And the Frisbees set up the foundation, which was quite active many years ago. And over the past almost 40 years, they've given over $3.5 million to the medical school, to the hospital, to the nursing school, to Department of Lab Medicine. And they have also funded this lectureship, which is funded now through the 2030s as their way of giving back and keeping Richie's name alive. So I wanted to, unfortunately, they can't be here. Normally they come here each year, but because of a family emergency, they weren't able to do that this year. So I wanted to thank them and let people know the amount of effort that goes into keeping the Cancer Center running and all the philanthropy that is so important to everything that we do here. So with that as an introduction, I will turn it back to Dr. Helene, and she will introduce our speaker.
All righty, we'll give you 10 minutes to speak, okay? Just keeping going back on introductions. Okay, so it's my absolute pleasure to now introduce Dr. Vijay Sankaran, who, let me just preface, you in for such a treat. He is the most amazing scientist, physician, and also human being. So again, you are in for a treat, and plus, nothing more beautiful than blood. So Vijay got his undergrad at Penn and then did his MD PhD residency and fellowship training in pediatric hematology oncology at Harvard and Boston Children's. He is the Jan Allen, is it Paradise professor of pediatrics at Harvard Medical School. He's a Howard Hughes Medical Institute investigator. He runs a phenomenal lab, but he also sees patients. And not surprisingly, he has won many awards. I asked him for a CV. It was 57 pages, so I didn't print it. But he has won the 2019 Selden Smith Award for Pioneering Research from the American Society of Clinical Investigation, the Meade Johnson Award from Society of Pediatric Research, the Paul Gallen Trailblazer Prize from the Foundation for the National Institutes of Health. He's an elected member of the Association of American Physicians and of the American Society of Clinical Investigation. And you will hear science, but what's so phenomenal is when somebody can take, human genetics, link it to disease and link it to functional disease, and then also come up, build this amazing foundation for therapies. And for example, one amazing thing really is Vijay worked on identifying BCL11A as a regulator of hemoglobin, and now we have gene therapies for hemoglobin disorders, sickle cell disease, and thalassemia. So I think we're in for another treat. So welcome, Vijay, and come out to the podium.
Well, thank you so much, Stephanie, for that kind introduction. And it's just been an incredible pleasure being here, having such incredible meetings and learning so much from so many folks today. So very excited to be able to talk to all of you and tell you about some of our very recent work. But I want to contextualize this in terms of how we've been thinking about some of these problems over time and how and where we think this might be going. So I want to start here. So each of us is thought to have approximately 100,000 blood or hematopoietic stem cells that reside within our bone marrow and that are responsible for maintaining and replenishing our blood and immune systems throughout our lifetime. This group of stem cells needs to produce millions of platelets, red cells, and a variety of white blood cell subsets every second in order to maintain our health. And this happens through this well understood process of hematopoiesis, which is depicted here. Now, arguably, hematopoiesis, as shown here, is one of the best understood examples of differentiation that we know of in physiology. We understand a great deal about the molecular regulators of this process, as well as how it occurs descriptively. And yet, when it comes to taking observations that we make in many of our patients, we often have challenges in mapping those observations onto our current understanding of this process. And so we've thought about that as not a limitation, but rather as an opportunity to take advantage of observations that we make from phenotypic variation in humans to be able to better understand how we can learn more about the process of hematopoiesis. And as Stephanie mentioned, our interest in this really started off about 22, 23 years ago. And at the time, we were interested in trying to understand why the most common monogenic disorders in the world, including sickle cell disease, is depicted in this electron micrograph, as well as thalassemia, both disorders due to mutations in the beta hemoglobin molecule, had such variable clinical severity. We knew there were patients who could present with actually very asymptomatic clinical courses, and on the other end of the spectrum, there were patients who were frequently hospitalized, required extensive care and extensive intervention. Now, we were curious what underlied this variation, and we had known from decades of work from a number of giants in our field that variation in the expression of a fetal form of hemoglobin could actually affect the likelihood of having clinical severity. So higher fetal hemoglobin could ameliorate the severity of both sickle cell disease and thalassemia. And it had been known that during human development, there is a switch from this fetal form of hemoglobin, or gamma globin, as shown in the green, to this adult form of hemoglobin, or beta hemoglobin, shown in the purple. This switch occurs shortly after birth, and the persistence of gamma globin, or fetal hemoglobin, can ameliorate symptoms in both sickle cell disease and thalassemia. And yet, despite the clinical observations that were known for a long time, for decades, the molecular regulation of this process remained largely unknown. And so, somewhat naively at the time, we said, well, maybe we could try to understand something more about this process. And so, after lots of failure, we were fortunate because after years of trying to work in the system in different model systems or different cell models, we were fortunate to be involved in some of the earliest studies looking at inter-individual variation that affected the level of fetal hemoglobin. And we had identified variation both in non-anemic individuals, and this was work that was initially done by Suile Etienne and the late Antonio Cao. And we were fortunate to be able to replicate this in one of the first cohorts with sickle cell disease. And these studies revealed 3 regions of the genome at the time that were associated with inter-individual variation in fetal hemoglobin levels, including this variation within chromosome 2 in the BCL11A gene. And that led us and others to identify BCL11A as a key and direct repressor of the gamma globin gene, such that when it's well expressed, there's repression of gamma globin, and the red blood cells that are produced, which is happening in all of your bone marrow cells right now, is predominantly expressing the adult form of hemoglobin. And at the earlier stages of development, we could show that there was lower BCL11A protein expressed, and this thereby de-repressed the fetal form of hemoglobin, allowing predominant expression of that. And this then led to lots of subsequent studies to identify how this gene was regulated, how we could take advantage of it. And as a result of an extensive amount of translational and clinical efforts, this has now led to the development of therapies targeting this axis, particularly the development of Exa-cel or Casgevy, which targets a erythroid enhancer of BCL11A using CRISPR-Cas9 genome editing in blood stem cells, thereby reducing the expression of BCL11A and thereby de-repressing fetal hemoglobin, resulting in amelioration of disease symptoms. And a number of clinical studies have been done to demonstrate the effectiveness of this, including studies that have been done in sickle cell disease, where individuals who, with sickle cell disease, who, as shown in this particular graph, had had frequent acute painful episodes, as indicated by the blue diamonds, after infusion of the modified stem cell product, actually could go on, as shown in the green bars, to largely have amelioration of their symptoms. And while there are patients who continue to show some symptoms, as indicated by the few blue diamonds that are shown overlapping the green bars, many of these patients have actually gone on to do incredibly well. And I've been very fortunate to actually, about a year ago, have the fortune to meet patient one in that study. This is Victoria Gray. And having a chance to talk to her not only revealed the fact that that the clinical symptoms could be ameliorated by such therapies, but actually her life has been transformed as a result of this. She has 3 children. She had a hard time caring for those children and actually could travel to see her husband, could do many more things with their children after receiving this therapy. So really hearing these stories has been incredibly inspiring for a lot of the things that we want to do. And so this early success has really motivated us to think about ways that we can more broadly think about how human genetic variation could teach us about the process of hematopoiesis, both in health and in disease, and where there might be opportunities. And so the 2 vignettes I want to tell you about today really exemplify this kind of approach. So over the past decade, there's been an increasing appreciation for the fact that while we think about blood and immune cell production as a stereotyped process, that we know that there's tremendous variability in this process, and that with aging in particular, individuals can acquire blood cancer driver mutations, often in the absence of an overt malignancy. And this is a condition that's been termed clonal hematopoiesis of indeterminate potential, or CHIP. And this is known to have an increased likelihood of progression to blood cancers, including myelodysplastic syndrome, as shown here, or acute myeloid leukemia, but does not invariably lead to these conditions and can also increase the risk for a variety of other comorbidities, including things like cardiovascular disease and a number of age-related complications. But when this condition was first described, it was thought that this was inevitably a pre-malignant condition that at some point would progress and evolve. But more recent studies, such as this study from George Vasilio's group, have revealed that there's actually tremendous variability. So even individuals with the same exact driver mutation can have tremendous variability in their likelihood of both having clonal growth, as shown on the x-axis here, as well as having progression to a blood cancer, such as acute myeloid leukemia, as shown in the y-axis. And so even patients with the same exact driver, such as in DNMT3A, they could show no growth or regression of their clone over time, or they could show some substantial growth. And that seems to be correlated with their risk of getting a blood cancer. And yet, this is not predictable simply based upon the mutations. And so that, of course, raises the question, what underlies this tremendous variability, and how can we better understand it? And studies from a number of groups, both observational and interventional studies in humans and model systems, have revealed a number of factors underlying some of this variability. We know that chronic infectious exposures, as well as inflammation, can underlie at least some of the variable growth rates that are seen in different clones, particularly with certain types of mutations. And so at least in part, this seems to underlie some of the variability. Certainly, some types of mutations, particularly those that are involved in DNA damage repair, such as TP53, CHEK2, and PPM1D, seem to expand in the setting of exposure to chemotherapy or radiotherapy, particularly in individuals being treated for solid tumors. But we wondered, what about the role of inherited genetic variation? And could there be a role for some of the factors either in increasing risk or protecting individuals? And so this caught the attention of a fellow in our group, Gaurav Agarwal, and Gaurav wanted to try to take advantage of a number of population-based studies that had been done previously and others that had not been, and integrate these together to really do a large genome-wide association study meta-analysis for both risk or protection from clonal hematopoiesis of indeterminate potential, or CHIP. But Gaurav wanted to particularly focus on those factors that were underlying variable risk of progressing or acquiring one of these clonal disorders. And so when he did this and examined different variants by their risk or ability to protect from clonal hematopoiesis, he was particularly intrigued by the most protective variant that we identified on chromosome 17. Now, this was a variant that, in a dosage-dependent manner, as shown on the top here, protected individuals from acquiring clonal hematopoiesis. So having 2 copies of this haplotype actually reduced the risk of clonal hematopoiesis by about 30%. And this occurred irrespective of the underlying driver mutation. So whether you had a DNMT3A, TET2, ASXL1, or JAK2 mutation, this variant and the presence of this variant seemed to be protective against all of these disparate driver mutations. But more importantly, this variant was also protecting individuals from acquiring myeloid malignancies, particularly acute myeloid leukemia and the myeloproliferative neoplasms. In addition, if we looked at other phenotypes, this variant was subtly but significantly associated with reduced blood counts from a variety of different blood cell subtypes, including platelets, a variety of white blood cell subtypes, including neutrophils and monocytes, as well as a reduced red cell count. But globally, this variant was not associated with other phenotypes as far as we could tell. So there was no additional exposure to any other diseases as far as we could tell, nor was there any impact beyond the impact on blood counts and protection from blood cancers and clonal disorders. If we looked globally, this variant was actually found across a range of different populations and was typically maintained at lower population frequencies, typically in the few percent range or lower in most populations, as shown here. So what was this variant doing, and how could we better understand what was happening there? And so as we began to look further at this variant and examine what was happening, we fine-mapped all the variants at this locus and could identify a single variant that overlapped a region of accessible chromatin that seemed to be a regulatory region in blood stem cells and other progenitors. This was a region in an intron of this gene MSI2 or MSASHI2, which I'll come back to, and was also bound by a variety of key hematopoietic stem cell transcription factors, including GATA2 and some of its cofactors, such as LMO2, LBD1, and so forth. This region of chromatin accessibility was actually selective for blood stem cells and some very primitive progenitors, but then actually became inaccessible as these cells differentiated. And in fact, there are no blood cancer cell lines that actually harbor this region of accessibility. So it only could be studied in the context of blood stem cells. And this region also showed long-range chromatin interactions with the promoter of the MSI2 or MSASHI2 gene. If we looked across the region harboring this variation, we saw that MSI2 was the only gene expressed in blood stem cells, and there was no other genes expressed in this region. If we looked at single-cell data from blood, stem, and progenitor cells of humans, we saw that there was a correlation between the degree of accessibility at this variant and the expression of MSI2, really suggesting that this was acting as an enhancer. But to directly prove that, we actually could use CRISPR interference where we could repress regulatory elements in primary human hematopoietic stem and progenitor cells. And when we do this and introduce these constructs into primary blood stem cells, we could repress selectively the expression of MSI2, but do not get repression of other genes in this locus, really suggesting that this was specific. Now, a number of groups have done extensive work to study the role of MSASHI2 or MSI2 as a key RNA binding protein that affects both the translation and stability of a variety of mRNAs in the cell. And that includes work that's happened here and elsewhere from a number of groups. So we wanted to understand, however, beyond some of the studies that have happened primarily focusing on progression in cancer where MSI2 is known to play a role, what was the endogenous role of this factor in normal blood stem cells, and how could we understand the role of this variant? And so initially, to get at this, we took this regulatory element in an exogenous setting, introduced it upstream of the endogenous promoter of MSI2, and then introduced that into blood stem and progenitor cells, and simply asked what was the activity like and how was this variant affecting that activity. And indeed, we could show that this regulatory element, if placed upstream of the endogenous MSI2 promoter in an exogenous setting, in this case introduced by a lentivirus, into these primary stem and progenitor cells, showed enhancer-like activity. So this increased the activity of this promoter, but this was actually reduced by the protective variant that protected individuals from acquiring blood cancers or from clonal hematopoiesis. If we looked at this region, we saw that this variant was actually impacting a canonical GATA motif that's got bound by GATA factors like GATA2 in this particular case. And indeed, we could show that there was reduced binding of GATA2 in blood stem and progenitor cells based upon the allele present, such that the protective allele in heterozygous donors was actually protecting individuals or preventing GATA2 binding at this locus. And importantly, this was also associated with reduced chromatin accessibility at the enhancer or at this regulatory element. But we then wanted to perturb this element in an endogenous setting. And unfortunately, we could not precisely perform base editing in an endogenous setting using at least the existing base editors, but we could recreate microdeletions that recapitulated, at least in reporter assays, the effects that we were seeing upon recreating this particular variant. And so therefore, we created these small microdeletions of 100 and 300 base pairs. We could also directly target MSI2 itself and examine its impact on different populations. When we targeted this enhancer, we saw about a 20 to 30% reduction in MSI2 expression within the blood stem cell compartment. We also saw that by looking at intracellular protein levels using intracellular flow cytometry, that in this population there was a reduction in MSI2 levels, subtly by or to moderately by targeting the enhancer, and more profoundly by directly targeting and editing MSI2, which served as kind of a baseline for further experiments. But we then wanted to understand what were the phenotypes that would occur upon targeting MSI2 or this regulatory element of MSI2. And what we could see is if we took, for example, adult hematopoietic stem and progenitor cells, or even cord blood-derived hematopoietic stem and progenitor cells, which I'm not showing you here, we saw upon editing this enhancer that there was a reduced content, both phenotypically and as well as in the numbers of these long-term repopulating hematopoietic stem cells, as shown here in these graphs, upon culturing these cells for at least a couple of weeks or a week to two. But we also wanted to understand what were the consequences of this if we were to perform long-term xenotransplantation, in this case, upon transplanting these cells into immunodeficient and mutant mouse recipients that allow reconstitution of blood cell production in the human blood cell production in the context of these mice. And if we transplanted an equivalent number of cells, either upon targeting the enhancer or upon targeting MSI2 directly, we actually saw a reduced engraftment of human hematopoietic cells, as shown in this bar graph over here. We could see that there was a reduced content of all lineages that we could examine, including stem and progenitor cells, B cells, and other myeloid cells as well, as shown here. But interestingly, if we actually replated these cells, we actually saw that there was actually more myeloid colonies produced. And indeed, we could see a skewing in terms of the percentage of myeloid cells produced. And this is consistent with the idea that MSI2 or MSH2 is regulating the symmetric versus asymmetric differentiation decisions that stem cells undergo, where either they undergo self-renewal in a symmetric manner or they produce differentiated progenitors to give rise to myeloid progeny, and MSI2 is skewing this difference. We then wanted to better understand what were the molecular mechanisms underlying this protection that was seen. And so to get at this, we could examine what happens upon perturbation of this enhancer in terms of gene expression in mRNA levels using single-cell RNA sequencing. And when we looked at and compared the expression, we see a variety of mRNAs whose expression is changed, either reduced or increased. And most reassuringly, the most reduced gene in this subset was actually MSI2 itself. But then we coupled this in collaboration with Michael Karas's group with deep profiling of potential targets of MSI2. And we could do this using this hypertribe assay where we link MSI2 to a editing enzyme, an ADAR editing enzyme, and then we can turn this potential binding problem into a sequencing problem looking at RNA edits. And this allowed us to identify approximately 3,000 mRNAs that were putatively bound by MSI2. And so then we could integrate that data together to ask which transcripts were both reduced in expression upon targeting the enhancer, but were also putatively bound by MSI2. And this showed us globally that there was typically reduced mRNA expression or levels amongst those mRNAs that were bound by MSI2, and allowed us to really prioritize a group of about 200 mRNAs that were both substantially bound or edited by MSI2, but that were also reduced in their expression. And this included a number of known regulators of MSI2, as well as some key factors involved in blood stem cells that had not been associated as being key factors involved in MSI2 regulation. Importantly, if we look at these group of mRNAs, we see that in blood stem cells, these are amongst the most highly translated mRNAs within the blood stem cells. So these, by ribosome profiling, are actually those that are ribosome bound to the greatest extent. We also could see that in individuals with clonal hematopoiesis or other clonal expansions, such as this individual with TET2, that this group of transcripts were actually increased in expression in the mutant stem cells as compared to the wild type stem cells in the same individual, shown here, for instance, for TET2 chip. And we can also see that this group of transcripts, akin to MSI2, actually portended poorer survival in individuals with acute myeloid leukemia. But everything I've shown you so far is really reliant upon the effect of this variant in the context of normal hematopoiesis. And so you might wonder, what happens if someone acquires a clonal driver mutation? And does this impact the propensity of that clonal driver to actually expand? And so we teamed up with Alex Bick's group at Vanderbilt, along with one of his MD PhD students, Yash Prasad, and we examined individuals in the BioView repository who had detectable clonal hematopoiesis at multiple time points with a median of 6 years between the measurements. And we could look at annual growth rate of these clones. And what we identified is across a variety of different clonal hematopoiesis drivers, that typically there was a range of different growth rates as had been seen in other longitudinal studies. But the presence of even one copy of this variant actually reduced the clonal growth rate across all of these drivers. Not only that, but this was for individuals who have clonal hematopoiesis detectable at both time points. But if you have a single copy of this variant, it reduces your likelihood or increases your likelihood of having that clone disappear in comparing the first time point to the second time point. So you are much more likely to have that clone regress or disappear or no longer be detectable in that context. So we then wanted to ask, is this potentially something we could model using some of the genome editing approaches we had taken? And I'll just give you one example of this, where we could recreate one particular driver mutation, in this case, truncating mutations in ASXL1, and we could simultaneously do this in the setting of creating either an enhancer deletion or targeting MSI2 directly. When we do this in vitro, we can see that where typically ASXL1 drives expansion of the phenotypic stem cell compartment, this is reduced by either perturbation of the enhancer or completely abrogated if we target MSI2 directly. Importantly, if we then do colony replating assays, if we target the enhancer, we actually can prevent ASXL1-driven colony replating. And if we also perform transplantation of these cells into immunodeficient, and in this case, KIT mutant mouse recipients, we actually can see that where typically there's an expansion of this very primitive CD90 positive compartment, this is reduced by targeting this enhancer. And so this and other evidence that we have really led us to a model whereby what we've learned from the naturally occurring human genetic variation is the fact that variation in MSI2 levels within blood stem cells seem to be a critical target in mediating both the overall pool of stem cells that's likely to be subject to acquiring somatic mutations and makes it less likely that these can acquire a driver mutation. But in addition, even upon acquiring a driver mutation, there seems to be a reduced likelihood that these clones will then expand. And in fact, in many cases, this makes it more likely that we'll see regression in this case across a spectrum of MSI2 levels. And really suggests, I think, to us and gets us excited about the idea that perhaps by learning more about this axis and targeting this axis, that we might not only be able to learn more about why individuals acquire clonal hematopoiesis or acquire blood cancers, but think about strategies that might allow us, as shown in this poster from Paris in 1932, to actually think about preventing blood cancer and actually fighting back and preventing individuals, particularly those at high risk. And so ultimately, I hope that these insights can start to at least inform us about how we might go about that or what the right strategies are. I don't have time to discuss it today, but I would just mention in passing, and I've included a QR code for our preprint on this, that we don't believe that this is the only mechanism that protects individuals. And in fact, we have evidence that there are other genetically inherited mechanisms that actually can protect individuals from acquiring clonal hematopoiesis. In this case, identifying a variant that protected individuals both from myeloid malignancies and some clonal expansions, particularly loss of chromosome Y, actually uncovered a long-known coding RNA that seems to play a critical role in HOX regulation and HOX gene expression. So certainly, I think that there are other mechanisms by which we can start to understand that. Now, I've given you an example of how human genetic variation can teach us about how individuals can be have variable risk of acquiring blood cancers as well as other clonal disorders, and really how we can learn about some of the fundamental mechanisms through the vantage point of human genetic variation. But in the second part of my talk, I want to switch gears a little bit and think more about how we can harness genetic variation to think about therapies. And while I've started with the example of how learning about BCL11A has enabled a variety of therapies targeting stem cells, enabling cures, and we know from an extensive amount of work that's been done in the field of gene therapy and gene editing that this has enabled curative therapies for a variety of blood disorders, immune disorders, and other sorts of disorders, largely now by using ex vivo approaches shown here on the left side. But increasingly, and I think that there is a possibility that these approaches might be transferable to in vivo approaches. But as we think about these approaches, certainly they're incredibly valuable, they're important to develop, and they're things that we and others are thinking a lot about. But could we start to take advantage of some of these developments to not only think about ways we can treat disease, but actually ways we could potentially prevent disease? And I want to give you at least a vignette today about how we might think about this and where there might be opportunities, and particularly where we could develop tools to enable this. So one disease that's incredibly prevalent, we know, is ischemic cardiovascular disease and ischemic cerebrovascular disease. We estimate affect about 40 million people in the world each year, with hundreds of millions of people affected. And our typical way of intervening in these contexts, whether somebody has an ischemic heart attack or stroke, is really to use a number of antiplatelet therapies, particularly aspirin and clopidogrel, as mainstays of this therapy. And these are incredibly effective and have been incredibly valuable. And yet we know compliance can be a challenge, as well as the fact that there can be individuals, even if they're optimally treated as shown here, who continue to show high platelet reactivity despite being on their medications. So patients can show either resistance to these medications or not be optimally anticoagulated even in the setting of some of these interventions. And this can actually increase the risk that a patient goes on to develop a secondary ischemic event. And so this got us excited about the idea, could we actually think about potentially in the future, with the development of a lot of these approaches and the advances that are happening in manipulating hematopoietic stem cells, could we, rather than thinking about these sorts of medications, in the future think about ways we could develop one-time therapies that could prevent platelet aggregation and yet allow for the possibility of reversal? And would this be a possibility? And you might at this point say, well, Vijay, go back to what you're focused on. You know a thing or two about hematopoiesis, and that's what you're focused on. That's what you should really think about. But we got excited about the idea, could we actually take advantage of durable but reversible manipulations in hematopoietic stem cells that could allow for antiplatelet therapies? And one particular advance that we got really excited about was advances from Angelo Lombardo, Jonathan Weissman, and others that have allowed for the development of epigenome editors. markers. And so initially, these really focused on modifying histones using crab domains recruited typically by Cas9 to specific genetic loci that enabled repression. I showed you an example of this earlier in the first vignette I talked about. But more recently, this has led to the recruitment of DNA methylation enzymes, including DNMT3A, either directly as part of the construct or indirectly by using histone tails that are from the histone H3 that then allow recruitment of endogenous DNA methyltransferases. And using this kind of approach, one can actually introduce DNA methylation selectively and in a targeted manner at specific loci. And so this has been applied across a range of different tissue types, including liver cells, neurons, as well as some cell models. But we really wanted to understand, could this work in the context of primary human hematopoietic stem cells? And could this be durable such that the progeny of these stem cells would actually retain the methylation once we had targeted this? And so this caught the attention of Chen Yi, a postdoctoral fellow in the lab, and she wondered, could she just get this approach to work in primary human hematopoietic stem cells? And if that was the case, could we actually apply this, such as in the setting of preventing platelet aggregation? So at first, we targeted for convenience a nice cell surface receptor, in this case, the don't eat me receptor CD47. And as you can see here, if we target this to normal human hematopoietic stem and progenitor cells, we, using RNA delivery, introduce these constructs into the stem cells and then examine the cells over time. What we could see is that with CRISPR interference, we could transiently inhibit the expression of CD47, but within about a week or two, this gets re-expressed within this compartment of stem and progenitor cells, whether we're looking at the more finely sorted group of stem and progenitor hematopoietic stem cell enriched populations or the bulk population. But importantly, if we look at either CRISPR off, which has DNMT3A as part of the construct, or CHARM, a more recently developed approach that actually uses an endogenous histone tail to recruit endogenous histone DNA methylases, we could see that there was sustained and durable repression of these targets. Concomitant with this, by bisulfite sequencing, we could actually see recruitment selectively of DNA methylation to the CD47 promoter in a targeted manner. And this was accompanied by repression of the mRNA of CD47 as shown here, really demonstrating that we could take advantage of this approach. And finally, we could also show that if we take this group of stem and progenitor cells and at least in vitro differentiate them towards different lineages, in this case, the red cell or megakaryocytic lineages, that we could actually retain the silencing despite the differentiation and despite all the global changes that happen in DNA methylation. So it started this off by mentioning how we wanted to take advantage of the fact that we could potentially inhibit platelets using this kind of approach. Hopefully, please don't fall asleep yet, because but with the dark. But so we reasoned that human genetic variation and human genetic evidence could actually give us a good sense of potential targets to go after. So as an initial target, we really wanted to take advantage of GP2B3A, a key target that we know from Glanzmann thrombasthenia is an important target, and that is the target of a number of therapies, such as abacizumab, that actually at least transiently and in an acute setting can prevent thrombosis. And so we just wanted to ask a very simple question. Could we inhibit GP2B3A in a selective manner within blood stem cells using the CHARM approach, then take the cells, differentiate them to produce platelets in culture, and just ask, does that prevent aggregation of these cells? And so, as you can see here, we could prevent or repress ITGB3, one component of GP2B3A, and using CHARM. And this is marked or is equivalent to CD61 when we're using this different antigens. And importantly, we can see that the majority of cells show repression of this. And critically, if we take these cells and differentiate them towards megakaryocytes where we're looking at the expression, we can see that if we look at RNA sequencing data really this is only selectively affecting the expression of ITGB3 and no other gene. So we're only seeing selective impacts just on the targeted locus. If we conduct whole genome bisulfite sequencing, in this case in either the stem or progenitor cells or in differentiated megakaryocytes, we see that the ITGB3 locus is really the only locus where we're seeing major DNA methylation across a region of about 1.5 kb, the CPG island that's present, and really across no other major regions. There are a few other blips that you'd note there, and those I would just note are typically small single CPGs or limited areas that are affected, and typically there we see no spreading whatsoever. And so here, for example, is just a plot to show you across different experiments where we could see that typically this not unmethylated region in controls at the ITGB3 promoter gets nicely methylated, and this is retained across these different divisions. So then could we actually get normal megakaryocyte, normal platelet production? And so with Kelly Macklis's group, we could look across megakaryocytes, and we could see that they were morphologically retaining all of their other features, with the exception that they would not express any CD61. And importantly, at least in vitro, we could form proplatelets that show appropriate morphologic features, but once again, do not have any expression of ITGB3 or CD61. And this is quantified here, where you can see that we can actually repress and limit the number of ITGB3 positive cells. But what about functionality? And so we could take these culture-derived platelets, which we know often behave suboptimally in comparison to those that we produce in our blood. And so we could actually mix normal healthy donor platelets with these culture-derived platelets, and then ask, upon simulation with different agonists, what is the ability of these culture-derived platelets to aggregate with healthy platelets? If we look in the control conditions, if we use ADP plus a thrombin peptide, we can typically get great aggregation. Similarly, with this thromboxane agonist, with ADP, we can get very good aggregation. And that we can completely abrogate by simply targeting with charm ITGB3. So we could prevent platelet aggregation in that setting. You might then ask, well, what about the durability of this approach? We're introducing DNA methylation, want to get erased over cell divisions. And so if we look in the primitive fraction of stem and progenitor cells marked by CD90 positivity and CD45 RA negativity, if we label these cells with CFSE to look at dividing cells, even amongst the dividing cells, we actually see that there's no erasure of ITGB3 silencing. This is maintained, and DNA methylation is maintained in these cultures. These, of course, are limited in terms of how long we can maintain stem cell and progenitor cells in culture, but we can use more recently described cytokine-free and chemically defined conditions that Hiro Nakauchi and Yamazaki have described. And indeed, even over this longer period of time, over several weeks, that we can maintain these stem and progenitor cells in culture, we can maintain DNA methylation across these time points. We can then subject the cells to differentiation towards the megakaryocyte lineage, and once again see maintenance of repression of ITGB3 or CD61 in this setting. And finally, we can also in vitro perform colony replating, where progenitors that would grow to reproduce colonies would need to undergo self-renewal. And we could do this across, in this case, tertiary colony replatings, and maintain the DNA methylation present in this setting, once again showing that we can maintain this targeted DNA methylation despite it not being there in an endogenous setting across multiple rounds of self-renewal. But really the ultimate test for us is, could we take these cells where we've introduced DNA methylation, introduce them into mice that are immunodeficient and have a KIT mutation? In this setting, when we introduce DNA methylation, we can actually get nice engraftment comparable to our control, so no difference in any human lineages that are detected. But importantly, we retain across 4 months post-transplant the DNA methylation present. And if we take those rare progenitor cells and then differentiate them towards either megakaryocytes or platelets, we see largely that the repression of ITGB3 or CD61 is maintained, and this is largely seen here. What about the safety of this approach? Could we reverse this approach in a targeted manner? And indeed, if we introduce DCAS9 with a TET enzyme, we can actually target that to these loci after introducing the methylation targeted to the ITGB3 locus. And if we, in a targeted manner, use a guide RNA to recruit that DCAS9 TET enzyme to the same locus, we can actually demethylate it If we then take those cells and differentiate them towards the megakaryocytic lineage, we could re-express CD61 upon methylation and demethylation and differentiation, really showing that we can restore expression in this setting. So you might say, okay, that's all well and good, but IgGB3 might not be the best target. What about the risk of bleeding? And I care for patients with Glanzmann thrombocytopenia. And we agree. I think that certainly maybe not the ideal target, but what about additional targets that are genetically nominated? And so we decided to go after a few additional targets. In this case, we initially targeted GP1BB, a key surface protein binding to von Willebrand factor. We could target this phospholipid scramblase ANO6 that's involved in platelet activation. And we could also target this other collagen co-receptor, SERPINH1 or HSP47. What happens when GP1BB is targeted? Well, we can nicely repress its expression. We could repress its surface expression as shown here. And critically, if we add ristocetin to these culture-derived platelets and look at their mixing with healthy donor platelets, we can see that while normally there's effective aggregation upon introduction of ristocetin, we can actually abrogate this binding by targeting GP1BB. Similarly with ANO6, which is this phospholipid scramblase, we can detect phospholipid on the outer surface of the platelets. And importantly, where typically if we use a calcium ionophore, we can get nice exposure in these culture-derived platelets of phosphatidylserine. We actually prevent that by targeting ANO6 in this particular case. And finally, for this other factor, SERPINH1 or HSP47, we knew about this not from human genetics, but rather from the fact that if you look at the differences between hibernating bears and non-hibernating bears, they seem to be protected from thrombosis by variation in platelet expression of this factor. And this is also seems to be the case in immobile humans. And so we decided, could we target this? And indeed, we can nicely repress SERPINH1 in this particular context using CHARM once again. But importantly, where typically we get a mild or moderate amount of platelet aggregation upon introduction of collagen plus ADP, we can actually prevent that upon targeting this particular region. And so once again, I think just giving you an example of where we see this going is with advances that we have in our ability to manipulate these cells. Our hope is that this and other kinds of approaches that we're taking will not only allow us to think about targeting blood stem cells for the context of blood diseases, immune diseases, and many of the diseases that we often are working on treating, but really try to extend the reach of these types of therapies, hopefully beyond this. And our approach and our motivation in thinking about this is how we can take advantage of human genetic variation to really nominate and ultimately manipulate this kind of process. And so this is, I hope, giving you just a flavor for where we see ourselves going and where we are excited about the future. So with that, I just wanted to acknowledge the people who contributed to this work, particularly Gaurav Agarwal, who contributed to the first story, first vignette I had a chance to tell you about, and Tenyi Yi, who contributed to the second vignette. We've had a number of collaborators on both of these stories, and our funding sources are listed there. And I especially, though, wanted to thank you all very much for both your attention, your willingness to come out here today. And I am happy to answer any questions. I'm very grateful to the Frisbee family for sponsoring this lecture, and just really grateful to Stephanie and all of you for the invitation. Thank you.
For questions, I'm going to take the first one since I have the microphone. Why is that variant so rare in Musashi? What's the benefit?
Yeah. Well, I think, what we've at least been thinking about is interesting, right? Because it's got an ancient origin in that it's found widespread across human populations. And of course, human populations are pretty recent, but there must have been reasons that it was maintained at a relatively low level of a few percent. What we suspect is that there's probably detrimental features that have happened in human history that might not be the case now, such as exposure to infectious agents. We don't know, and I think that requires further exploration. But we have been looking, at least in modern population biobanks, there does not seem to, as far as we could tell, be any detrimental phenotypes associated with it. I think we need to explore further, but that's at least our speculation.
Super, awesome. Mina? Just turn it on.
really amazing story about the germline resilience to CHIP. One question, you kind of mentioned it a little bit here, was how is it not correlated at all to cardiovascular disease? And the other is there is a part about myeloid skewing for those patients, and there is, I guess, not too much data right now about its correlation to kind of an aging bone marrow phenotype.
Yeah. Yeah. Well, so to answer your first question, I think where we've looked at, I think we've been underpowered with the effects it has on CHIP and thereby cardiovascular disease. But at least if you look at the direction of effect, it does seem that it's not acting in a different way. So it's not saying, I don't think that there's any indication that it's protecting against acquisition of CHIP but not protecting against cardiovascular disease. And in fact, if we look at the general direction of effect, I don't think that the effects are significant, partially because of power issues, but we're looking at broader populations. I do think that at least the evidence we have suggests that it likely protects against at least CHIP-mediated cardiovascular or CHIP-associated cardiovascular disease. In terms of what it's doing to aging subsets, I think that that's such an interesting area and one that we're excited to explore, because I've sort of given you the Richard Scarry version, which is, not all blood stem cells are the same. We know in our field, we're thinking a lot about heterogeneity of these different populations. And so I think that there's a lot more to explore about, how is this variant impacting those different subsets perhaps differently, and how is that modulating the age-related impacts that we might have? I also think to that end, does this protect against other age-related morbidities that are also at risk for CHIP? And I think certainly we're hoping to look at some of those cases. We don't have great evidence right now. That's great.
Vijay, that was fabulous. Thank you. From your slide, I thought that the distribution in the population of this resilience variant was highest in the Finnish population. Is that real?
Well, it's marginally so. So in the Finnish, it's about 6%. And across various European populations that we've looked, it's typically in the range of 4 to 6%. So I think some of that's probably biased just by the way I plotted it, because you're looking at differences in some Asian populations of 1% or slightly under that versus 4 to 6%. It's been maintained at relatively low percentages, I think, across all populations, but certainly some populations like the Finnish that have had a bottleneck might have higher frequencies. We don't think that there's necessarily, at least as far as we can tell, like really selection in one population versus another. But I certainly think, to Stephanie's earlier question, there's probably a lot we have to understand about what happened in human history, why this was maintained at such low frequency.
So are there more questions? Well, I have more questions. Not a lot of grand rounds that cover both malignant and classical hematology. So how many stem cells do you have to modify in a person to get an effect on the platelet aggregation that's clinically then meaningful?
Yeah, it's a really good question. And I think, one limitation, as you know, about these humanized models is they don't give rise to platelets very well. And so we're going back and doing some of this in mouse stem cells, with the goal that we can do laser-induced thrombosis and other types of things. But I do think if you were to get, we're doing all this ex vivo with transplantation. So, I think ultimately, if you were to manipulate and be able to get in vivo even most stem cells modified, I think this could be beneficial. But I certainly think that that's where we'd like to look across multiple targets. We'd like to kind of play around with these sorts of approaches. Really, this came about in part because we were just looking at, could we even do this? Would the methylation be sustained? I think this was a good target to go after. And I'm not necessarily saying like, we're going to, because I still think, for most of us, aspirin and clopidogrel or other antiplatelet agents are going to be a mainstay for a long time to come. But certainly I think it does suggest that perhaps we need to think about broadening this. And our thought is, we could get good, approaches with all the produced platelets, even after xenotransplantation, and prevent aggregation by targeting these axes. So I do think you'd have to target the majority of stem cells to really get this effective, but
it's phenomenal, right? Because you could also potentially modify the bone marrow microenvironment and then create an environment that is less inflammatory, less conducive, for example, than to clonal hematopoiesis.
Exactly.
yeah.
And in fact, Ten Yi, who went into this, she sort of said, "Oh, I'd like to make aging-related changes." And I said, "Those are going to be complicated. You're not just going after one loci. Let's actually show, what can be done across all these different loci as a starting point." So,
Fantastic.
thank you.
Hello. Oh, hi. Thank you for a wonderful talk. Along similar lines to Dr. Helene's question, how translatable do you think that technology is, and what is the time scale for the reversibility? Do you think this is something that's clinically kind of applicable, and in what other contexts do you see this maybe being used?
Yeah. So I will say, and I think, as you can imagine, we initially set out for some other targets. It turns out the BCL11A locus is actually methylated in stem cells, so it's not a good target for such methylation-based approaches. I think that there are other colleagues in the field who've targeted other regulators of fetal hemoglobin, and certainly it works there. And so I think that probably some of those applications might happen earlier. It's a little hard to know because I think that really, that's where we're interested in showing that this works and thinking about broader applications. I'm not sure antiplatelet therapies are going to be the application tomorrow, but I certainly think that there might be, excitement and interest if we could find the right target, and maybe this is or isn't the right approach. But in terms of, delivery and using this actually is no different than what's currently clinically done for, Cas9 genome editing or other types of editing reagents that are being delivered. It does differ a little bit because we're using mRNA delivery, whereas some of the approaches use ribonucleoproteins and proteins and things of that sort. But I think ostensibly, really, that's where we hope if, different companies either get the ex vivo or even in vivo approaches optimized for different mRNA delivery, these payloads could easily be, targeted. I think one attractive feature also about the DNA methylation is you can actually multiplex it, and we've done that in some cases. I'm not sure for the applications we show that's what you'd want to do, but certainly I think that in the future you could think about, are there ways you could augment stem cell engraftment at the same time that you are, or reduce inflammation in a stem cell at the same time that you're also, repressing a target that could prevent thrombosis, et cetera. So.
Okay, phenomenal. So if there are no more questions, doctors, done.
Could you tell us, whenever you introduce gene changes into a population, then transplant into the bone marrow and hopefully to get what you wish to get, you will have a heterogeneous population with respect to that particular gene you're targeting on. What's the interplay?
Yeah. You mean in terms of the heterogeneity that we get, that we would...
The result of modifying a gene.
Yeah. I think it's definitely a limitation. I do think for a lot of what we've done, experimentally, one thing that's been of benefit for us is, especially with some of the base editors and other editing reagents we've had more recently, we've been getting very effective editing. And so, in those cases, we can, in many cases, get 90 to 95% of alleles edited. And so we think in the majority of cases, we're really getting both alleles in the stem cell, even where we've done single cell cloning. But I think you raise an even more sort of broader point, which is, as we think about these manipulations, how can heterogeneity play a role? How can we optimize this? And do we need to optimize this? And I think that's actually something we're really excited about as we've even gotten like the HSC methylation going, because as you can see, there's still some heterogeneity as we do these transplants. We get some cells that may be a little bit less methylated that seem to erase this. And so we're excited about ways we could both understand some of that heterogeneity, but also try to develop better reagents. And we're working with Jonathan Weissman to do this, where we could actually better methylate those regions, either by stabilizing some of these domains or targeting them to specific loci even more effectively. So I think heterogeneity is still probably a little bit of the bane of our existence, but also something we can hopefully work on and improve. But I already feel like for some of the editing we want to do, we're already getting really effective editing. So I think it's come a long way. Stephanie and I were talking earlier, and it's amazing to think, like, compared to 5 years ago, even what we can do with human hematopoietic cells. And so, give us a few years, but I just feel like, I think maybe not me, but, at least the younger people, there's lots to figure out, I think. There's lots to do, which is great. So.
I'm more or less thinking from the variation from the evolutional point of view.
Yeah.
You begin with something which altered. Eventually, in that particular individual or race, that variation become dominant.
Yeah.
End up with you have related either is to its advantage or to its disadvantage. This is coming back to what you're trying to manipulate the gene within the compartment. Here is the bone marrow.
Right.
Well, some way, I think the heterogeneity actually is maybe play a very important role in recognizing whether it is. In other words, you don't need to achieve homogeneous population.
Yeah.
The heterogeneity of the different population may play the role of eventually with the health issue you want to address instead of that's just a general comment.
Yeah.
Thank you.
Thank you.
I think we're soon running out of time because it's going to be the trainees' time with Dr. Sankaran. So thank you so much for this absolutely phenomenal lecture. And I hope you brought a bag because you're going to get a present.